Hi everyone, and welcome to our video on DNA sequencing. What we're going to be doing in this video is having a look at the specification reference 6.3.1a, the principles of DNA sequencing and the development of new DNA sequencing techniques. First thing, what on earth are we referring to when we talk about DNA sequencing? Well, quite logically, it's in the name. We're working out the sequence of nucleotides in a strand of DNA itself. So we are literally working out the sequence of the DNA, and DNA, hopefully as we remember, is a polymer made out of these monomers called nucleotides. Therefore, the sequence of nucleotides is our DNA sequence. If we jump back in time to 1972, then we are actually at the point that we first sequenced a whole gene. So in the grand scheme of science, this is pretty recent. Let's be honest, 1972 isn't that long ago, really. I know it feels like the ancient past right now, but if we consider the whole of the science history, then it's not, it's recent. And the first gene that we sequenced was for this thing called MS2, which is actually a coat protein for a virus. And the way that we actually sequenced this gene was using mRNA, the messenger RNA. And this is not a perfect way to do this, for the simple fact that RNA isn't a stable molecule. In its very nature, it's quite transient. And therefore, if we were trying to actually carry out sequencing of large genes using something like mRNA, it just wouldn't work because the mRNA would actually degrade before we'd sequence the whole thing. So it worked with MS2 because that was a relatively simple gene, relatively small gene, but it wouldn't work for these larger genes that we may wish to sequence. The first scientist that we really need to know about, and the first technique that is of importance to us, is one that came about back in 1975, and it was developed by the scientist called Frederick Sanger. Now, as usual in science, when scientists discover something, then the little bit of ego that I suppose many of them have means that they name it after themselves. So the technique that Sanger developed is what we refer to as Sanger sequencing. It does have a different name called chain termination method of sequencing, but that doesn't quite roll off the tongue to the same way that Sanger sequencing does. So what is Sanger sequencing? What was his method? What he actually did was he used this single strand of DNA as a template, and then he used these things called modified nucleotides, which have the name dideoxynucleotides. And what these modified nucleotides have is a little radioactive isotope label on them. So when we actually have our single strand of DNA, that's our template, these modified nucleotides with their little radioactive isotope tag on them are then going to pair up with their complementary base pair on the DNA template. Now, in order to carry this out, what we actually have are these 4 test tubes. So if you imagine just having 4 little test tubes all set up, then in each of these test tubes we are going to have the DNA is in all 4 of these. So in all 4 of these test tubes we will have our single-stranded DNA. So we've got a single-stranded DNA template in there. We have got our DNA polymerase, which hopefully we remember is an enzyme involved in obviously DNA replication. We have got these DNA primers. Now the DNA primer is necessary to allow the DNA polymerase to bind. And then we have our free nucleotides. So we have our regular nucleotides. And then in each of these 4 tubes we are going to have our dideoxynucleotide, and we will have one of each type. So in this first tube we would have our dideoxynucleotide for adenine, then we'd have our dideoxynucleotide for thymine, dideoxynucleotide for cytosine, and our dideoxynucleotide for guanine. So in each tube, we've got our standard mixture of the single-stranded DNA, the DNA polymerase, the primer, and the free nucleotides, non-radioactive labeled ones. And then we have one nucleotide that is the modified one with that radioactive tag. Once we've put all of those little components into our 4 test tubes, we then incubate them. Now, during this incubation process, what's going to happen is the primer is going to anneal, so it joins onto the start of that DNA template. So where we've got our single-stranded DNA, we then end up with a little bit of a primer, and therefore we've got a short section of double-stranded DNA at that point. That's the point that our DNA polymerase can then join on to our primer, and it will then begin DNA replication using those free nucleotides present in the tube. So basically our DNA polymerase is then just going to move along that strand, adding nucleotides as it goes. So once we've actually finished this incubation period, what we then do is separate these different length DNA chains from the template DNA. Now at this point you're probably thinking, well, why are they different lengths? Well, if you remember what we said, we've got this template strand of DNA. In the tube, we've got regular nucleotides, and then we have one of those modified nucleotides with a radioactive tag on it. So what happens is, as our DNA polymerase happily moves along, it's just joining the regular nucleotides, but if it happens to join one of our dideoxynucleotides on, that acts as a terminator. That means it's going to stop any more of that complementary base pairing further along. So we would end up with a relatively short strand here. In that same test tube though, on another template strand, we could have a greater number of our bases complementary base pairing up with the regular nucleotides before we actually get to one of our dideoxynucleotides being added, and therefore we've got a longer chain. So what we end up with is just down to that chance of when the modified nucleotide is added determines the different lengths of the chains. So once we've actually separated those from the template strand, we basically just have this mixture of different length DNA fragments, and what we're going to do is use gel electrophoresis to separate them out. So gel electrophoresis is where we've got this slab of gel, and what we have, these little wells at one end, you put your sample in there, and we obviously place it into our gel electrophoresis tank with the solution. And what we're going to then find is, as we turn on the electric current, the DNA is going to be moving towards the anode, the positive electrode. The reason for that, DNA has a negative charge on it. So because the DNA is negative, it's attracted to the positive electrode at the far end of the gel, and therefore it's going to move up the gel. Because we've got different length fragments, they move different distances. Shorter fragments go further. Bigger fragments don't travel so far. What Sanger then did was sorted out all of these fragments by their chain lengths and read those radioactive labels. Now that did require a certain element of specialist training to be able to obviously interpret this radioactive little image that we got, but we got this little bit of information about which nucleotide was in the end point of each of those different length fragments. Now what we then did was we started off with the shortest fragment, and we were able to say, oh, that one has the modified adenine at the end of it. So our DNA sequence starts with our adenine. We would then move along and go, oh, the next one is a cytosine, and literally just moving along the chain, reading what that modified nucleotide was in each case, we were able to assemble that final DNA sequence. As you can probably already imagine, taking each individual fragment, working out the little order of them, and therefore which base is at the end of it, takes quite some time. Anything that's time consuming in science is also quite costly, and therefore we've got to have obviously quite an investment going into this process, both time and money. We also obviously require specialist staff to read those autoradiograms, because you couldn't just wander in and do it without any prior knowledge. So we need specialist staff who are going to be working for quite an extended period of time on this. Therefore, it's going to cost a lot. Obviously, this wasn't going to be the only method that we used, because once we'd started doing this sequencing thing, then people wanted to do it more, but they didn't want to be spending hours and hours and hours poring over these autoradiograms. So, 1986, we get our first DNA sequencing machine coming into existence. Now, the key difference here between Sanger and our DNA sequencing machine is, rather than using these radioactive tags to label those terminal bases, we use a fluorescent dye instead. Now, the advantage of that is, by using a fluorescent dye, we could then use a laser to scan our sample, and the computer will then interpret the information from that laser and produce the final sequence for us. So this is now moving to an automated process. Basically, the computer is able to read this for us, not someone sitting there painstakingly going one base at a time. So the way this works then is, each type of our dideoxynucleotide is labelled using a specific fluorescent dye. So they're different colours. So our adenine would be the green, the thymine the red, cytosine blue, and our guanines would be yellow, for example. Now, what we use is a process called capillary electrophoresis to then separate the single-stranded DNA by its mass, i.e. we're going to have it so that all those different length chains are separated out from one another. We then shine the laser across that capillary, and what we're going to find is they're going to illuminate the different dideoxynucleotides, and based on the colour and where it is in that capillary tube, we then get the DNA sequence. In terms of the advantages of using this DNA sequencing machine, it's obviously faster. It's clearly going to be able to do this on its own without the need for specialist technicians. If you can set it up, then it's going to do it for us. And another advantage in science, it's not using radioactive labels. And hopefully we know from a little bit of our science history, playing with radioactivity all day, every day in a lab isn't great for your longevity. A lot of people who spent time playing with lots of radioactive things in experiments in labs don't have a long life expectancy, because obviously the more you're exposed to radioactivity, the greater your own risk of mutations, i.e. developing cancers, will be. So, moving further towards the present day, we are now in 1996, almost in the current millennium. And what we developed here was this technique called pyrosequencing. Sounds far cooler than the others. Now, what we're doing in our pyrosequencing is we're going to actually cut the DNA that we're going to be sequencing into these short fragments of about 300 to 800 base pairs. And that's done using a special bit of equipment called a nebulizer. Once we've actually cut our DNA into these little fragments, we then degrade it into the single-stranded DNA. So basically we're splitting the double strands into the single. So that gives us the template. Now that template we are then going to immobilize on this thing called a flow cell, which to all intents and purposes is a little plastic slide. Once we've got our DNA immobilized on the flow cell, we incubate that in a reaction mixture which contains our primer, the DNA polymerase, and then some new players, ATP sulfolase, luciferase, apyrase, APS, and luciferin. Now, hopefully we can already pick out, just looking at the structure of these names, that because we've got a whole bunch that end in A's, these are different types of enzymes. What's going to happen then in our pyrosequencing? Once that mixture has all been incubated, the first reaction is going to take place by adding the activated nucleotides containing only one of those 4 bases to the flow cell. If the first base in the sequence is complementary to the one we've just added, then the nucleotide will be added to its chain by DNA polymerase. When that happens, what's going to result is the release of this diphosphate. Now, the diphosphate itself reacts with the APS from our reaction mixture, and that forms ATP, all catalyzed by our first enzyme in the mix, ATP sulfolase. So basically what we're seeing here is as our little base joins onto that actual sequence, we are then releasing diphosphate, which reacts with the APS to form ATP. Then the ATP is used by our second enzyme, luciferase, which then converts that luciferin in our mixture into oxyluciferin. And that's a reaction that is going to generate this little pulse of visible light. Now, this little pulse of visible light is going to be detected by a camera that's set up over our flow cell, and we then have our apyrase, our third enzyme, that basically degrades any nucleotides that are not incorporated into that DNA template. Next up, different activated nucleotide is washed across our flow cell. If it's incorporated, same process happens. We generate a flash of light that's picked up by the camera. And then we just keep repeating this with all of our different nucleotides until we have fully synthesized the whole DNA fragment. And what we'll have done is, at each point that one of those nucleotides bound, we generate that little pulse of light which the camera has detected. So what we're looking at in terms of our pyrosequencing is something that is cheaper, it's much faster, and because we're obviously looking at basically immobilizing it on this little plastic slide, we can sequence thousands or millions of DNA molecules at the same time. Hence why it's so much faster and cheaper than other methods. Now, pyrosequencing is one of these examples of our next generation sequencing. Sounds almost like a Star Trek thing. But next generation sequencing, or NGS, is where we have got a new method of DNA sequencing that has superseded the Sanger method. So we've replaced the Sanger method with something, it's called next generation sequencing. The next sequencing technique that we are hopefully going to have at our disposal is this one called nanopore sequencing. Now this is still in development, but when we've got it up and running fully, this is going to allow us to carry out the DNA sequencing outside the lab, so actually in the field we could do this, and it's going to be incredibly rapid. So this really is the future of our DNA sequencing, if you like. Now the way the nanopore sequencing works then is we are going to have basically a little device that we are then going to add a DNA sample to. What then happens is we have got this little thing called a nanopore set up, and what that's going to allow us to do is to move this little DNA molecule through the pore. So what's going to happen is our DNA molecule is going to be unzipped by the top of the pore, so that a single stranded bit of DNA is then going to pass down through this thing called a nanopore. And basically what we're going to then see is, as each of the bases is going to alter the flow of current in a very slightly different way from one another, what we're then going to generate is a slight change in current that can be detected as it moves through the pore itself. So basically what we're able to do is, by just recording the little change in current that's caused as the DNA moves through this little tiny nanopore, we can work out the sequence, because each base generates this own unique little signature of current flow, if you like. Now, when we are carrying out all of these sequencing techniques, what we are doing is generating vast quantities of data. So we've obviously needed to actually develop a whole new field of biology, if you like, called bioinformatics. Now, bioinformatics is all about storing, retrieving and analysing the data that we've generated through all of these biological studies. So it's looking at those DNA sequences, the RNA sequences, protein sequences, the relationships between genotypes and phenotypes. All of that is going together to give us this new field of how we can actually store and use this information we've now generated. When we're thinking about the databases that are going to store this information, they need to be pretty high power computers here, because this is vast quantities of information that we don't want some kind of a snail's pace computer like most of you probably have at school, where you type something and then you're waiting a few minutes for it to actually follow through on what you've asked it to do. You don't want that when we're talking about retrieving scientific data. So we need high power computers able to process this. So once we've actually sequenced one of these genomes for any particular organism, bioinformatics is going to allow us to compare that genome to different organisms' genomes already in the system. So it's going to obviously be comparing that entire DNA sequence to who knows how many other organisms. That requires a certain amount of computing power to do. In terms of what we use these databases for then, first thing is we need this bioinformatics system to give us access to large amounts of data. And this data is going to be on both DNA and proteins. So it will have DNA sequences, it's going to have protein sequences, and that's going to give us the ability to obviously do a lot of comparisons, but only if that information is held in a universal format. So it can't be that one lab stores it in this way and another lab stores it another way. The idea is to create this universal format storage drive, if you like, globally, of data about living things. Their DNA sequences, their protein sequences, so that all these researchers around the world can access it, they can run comparisons with anything they want to, they can analyse it in their own way for their own means, and obviously advance science from that. So if you're asked about bioinformatics, it's all about facilitating access to large amounts of data on DNA and proteins, and this information must be held in a universal format. As always, I do suggest you subscribe to the channel so you can see when I next upload a video, and of course head on over to the A-Level Biology website so that you can get access to a range of other resources, hopefully there to help you with your A-Level biology studies.